The very essence of life as we commonly perceive it is inextricably linked to the air we breathe, to the vital oxygen that fuels our cells. Yet the natural world, in its boundless ingenuity, presents us with creatures that defy this fundamental rule. Imagine, if you will, organisms that not only endure but truly flourish in environments devoid of this life-giving gas, realms where the familiar processes of respiration are simply an impossibility. These are not mere scientific curiosities. They are testaments to the relentless adaptive power of evolution, a force that sculpts life into myriad forms, each perfectly suited to its own unique, often hostile niche. These remarkable animals have, over eons, developed an astonishing array of strategies to navigate existence without oxygen. Some have forged symbiotic relationships, others have radically altered their metabolisms, and a few have even discarded the very cellular machinery that most life depends upon for aerobic respiration. Their existence pushes the boundaries of what we understand about biological possibility, offering profound insights into the flexibility and resilience of life itself. They inhabit the darkest depths of our oceans, the hidden recesses of the earth, and even within the bodies of other creatures, each a master of survival in an oxygen-starved world. Prepare to journey with us into these unseen corners of our planet, to encounter beings that challenge our most basic assumptions about what it means to be alive. We will explore the extraordinary mechanisms and the sheer tenacity of creatures that have conquered the breathless frontiers. You won't believe how these animals survive where nothing else can. Their stories are not just fascinating. They are a humbling reminder of nature's infinite capacity for innovation and endurance, a narrative written in the very fabric of their being. Our journey into the world of anaerobic animals begins with a truly astonishing discovery, one that rewrote the textbooks of zoology. Deep within the muscle tissues of salmon resides a microscopic creature known as Henneguia salmonicola. For years it was classified alongside other cnidarians, a group that includes jellyfish and corals, all of which it was believed required oxygen. However, meticulous research into this tiny parasite revealed something utterly unprecedented. It is the first multicellular animal known to science that can survive entirely without oxygen, a true outlier in the animal kingdom. The implications of this discovery are profound. Henneguia salmonicola belongs to a group called myxozoans, bizarre, spore-forming parasites. What sets it apart is not just its ability to thrive in anoxic conditions, but the very reason it can do so. Genetic analysis has shown that this creature has completely lost its mitochondrial genome. Mitochondria, often called the powerhouses of the cell, are essential for aerobic respiration, the process by which most animals convert oxygen and nutrients into energy. Without them, Henneguia simply cannot breathe oxygen in the way other animals do. Instead of relying on oxygen, it employs an entirely different metabolic strategy. It exists in an intimate relationship with salmon, forming small, whitish cysts within the fish's flesh. These cysts, sometimes referred to as tapioca disease, are the visible manifestation of an organism living completely independent of oxygen. The salmon, swimming through oxygen-rich waters, unknowingly carries a passenger that has abandoned the need for air altogether. The evolutionary path that led to this adaptation is a fascinating puzzle. Its ancestors likely possessed mitochondria and respired aerobically. However, in the stable environment within its host, the machinery for oxygen respiration became redundant, eventually being lost entirely. This is a remarkable example of reductive evolution, where complexity is shed for specialized existence. Henneguia demonstrates that the definition of animal can be far more flexible than we once imagined. Its discovery opens up a new frontier in our understanding of animal life. It challenges us to look closer, to delve deeper into the hidden corners of the biosphere, for who knows what other breathless wonders await discovery. The story of Henneguia salmonicola is a powerful reminder that nature's capacity for innovation is truly limitless. Plunging into the crushing pressures and eternal darkness of the Mediterranean Sea's deepest basins, far beyond the reach of sunlight and, crucially, oxygen, we encounter another group of extraordinary survivors, the Loricifera. These are microscopic marine invertebrates, no bigger than a grain of sand, belonging to their own distinct phylum. For a long time, it was assumed that complex, multicellular life could not persist in these permanently anoxic, hypersaline environments. The discovery of loriciferans not just surviving, but actively reproducing here, was a revelation. In the L'Atalante Basin, a depression on the Mediterranean seafloor over 3,000 meters deep, the water is so dense with salt that it doesn't mix with the oxygenated waters above, creating a truly alien, oxygen-free habitat. It was here, in sediment samples painstakingly brought to the surface, that scientists found 3 species of Loricifera: Spinaloricus, Rugiloricus, and Plissaloricus, living their entire lives without a breath of oxygen. Unlike other creatures found in anoxic zones, which might be temporary visitors or dormant forms, these Loriciferans were metabolically active, with evidence of feeding and reproduction. Their secret lies, in part, in their cellular machinery. Instead of mitochondria, the oxygen-dependent energy factories found in most animal cells, these Loriciferans possess organelles that resemble hydrogenosomes. Hydrogenosomes are found in some single-celled anaerobic organisms like certain protozoa and fungi, and they generate energy through processes that do not involve oxygen, often producing hydrogen gas as a byproduct. The presence of these structures in a multicellular animal was a groundbreaking finding, suggesting an ancient adaptation to life in oxygen-poor or oxygen-free conditions. The Loriciferans themselves are intricate creatures, despite their minute size. Their bodies are encased in a protective outer shell, or lorica, from which they derive their name. They possess a spiny head or introvert, armed with scalids, which they use for locomotion and likely for feeding on bacteria or other organic matter within the anoxic sediments. Their ability to complete their entire life cycle, from egg to adult in these extreme conditions, marks them as true extremophiles, pushing the known boundaries of animal endurance and metabolic versatility. The existence of these deep-sea Loriciferans not only expands our understanding of where animal life can thrive, but also offers tantalizing clues about the nature of early life on Earth, when oxygen levels in the atmosphere and oceans were significantly lower. They may represent a lineage that either retained ancient anaerobic pathways or re-evolved them to colonize these challenging, yet resource-rich abyssal plains. They are a testament to the persistent colonization of even the most inhospitable niches our planet has to offer, hidden architects of life in the profound dark. Perhaps one of the most famously resilient creatures on our planet is the tardigrade, affectionately known as the water bear or moss piglet. These microscopic invertebrates, typically less than a millimeter long, are found in an astonishing variety of environments, from the highest mountains to the deepest seas, from scorching deserts to frozen polar regions. While not exclusively anaerobic, their ability to survive periods without oxygen is part of a broader suite of extraordinary survival mechanisms that make them virtually indestructible, a true marvel of the natural world. Tardigrades can endure conditions that would be instantly lethal to almost any other known animal. They can withstand extreme temperatures from just above absolute zero to well over the boiling point of water. They can survive crushing pressures, exposure to intense radiation far exceeding human tolerance, and even the vacuum of outer space. Central to many of these abilities, including surviving anoxia, is their capacity to enter a state of cryptobiosis, a reversible metabolic suspension where life processes come to an almost complete standstill. When faced with adverse conditions such as desiccation or a lack of oxygen, tardigrades can undergo a process called anhydrobiosis. They retract their 8 legs, contract their bodies into a compact barrel-shaped form known as a ton, and reduce their metabolic activity to less than 0.01% of normal levels. In this ton state, their water content can drop to as little as 1% of normal. This profound dehydration protects their cellular structures from damage and allows them to remain dormant, sometimes for decades, awaiting the return of more favorable conditions. During anoxia-induced cryptobiosis, or anoxibiosis, tardigrades similarly shut down their metabolic processes. While the precise biochemical mechanisms are still being unraveled, it's understood that they can significantly slow down or alter their energy production pathways, surviving on stored reserves without the need for oxygen-driven respiration. Once oxygen becomes available again, or in the case of anhydrobiosis, when rehydrated, these remarkable creatures can revive, seemingly unharmed, within a matter of hours or even minutes, resuming their normal activities as if nothing had happened. The tardigrade's unparalleled resilience offers invaluable insights into the fundamental mechanisms of life and survival. Scientists are keenly studying their unique proteins, such as damage suppressor proteins, which appear to protect their DNA and other cellular components from extreme stress. Understanding how tardigrades achieve these feats could have profound implications for fields ranging from medicine, such as preserving organs for transplantation, to astrobiology, in considering the potential for life to exist on other planets. These tiny water bears are a colossal testament to life's tenacity. Our exploration now takes us into the realm of fungi, specifically to a single-celled organism that has played a pivotal role in human civilization for millennia, Saccharomyces cerevisiae, commonly known as brewer's or baker's yeast. While often overlooked in discussions of animal-like survival, yeast as a eukaryote actually shares complex cellular structures with animals, and its ability to switch its metabolic strategy in the absence of oxygen is a fundamental example of anaerobic adaptation, one that we have harnessed for our own benefit. Saccharomyces cerevisiae is what we call a facultative anaerobe, meaning it can thrive in the presence of oxygen, where it performs aerobic respiration much like our own cells. However, when oxygen becomes scarce, it possesses the remarkable ability to shift its metabolic gears. It switches to a process called anaerobic respiration, or more specifically, fermentation, a strategy that allows it to continue producing energy without oxygen. During fermentation, yeast cells break down sugars into pyruvate. In the absence of oxygen, the yeast converts it into ethanol and carbon dioxide. This process is less energy efficient than aerobic respiration, but it's a vital survival mechanism, allowing the yeast to continue to live and reproduce when oxygen is not available. This metabolic flexibility is precisely what humans have exploited for thousands of years. The carbon dioxide produced during fermentation is what causes bread to rise. The ethanol is the key ingredient in alcoholic beverages like beer and wine. So, an ancient survival strategy of a humble fungus has become a cornerstone of human culinary traditions. The study of yeast's anaerobic pathways has been instrumental in advancing our understanding of biochemistry and genetics. Its ability to navigate between oxygen-rich and oxygen-poor worlds makes Saccharomyces cerevisiae a fascinating example of life's adaptive ingenuity. We now venture deep into the Earth's crust, to environments that seem, honestly, inhospitable to any form of complex life, to find another group of oxygen-defying creatures, nematodes or roundworms. These ubiquitous, unsegmented worms are found in nearly every habitat on Earth, from garden soil to the deep ocean, and even as parasites within other organisms. What is particularly astounding is the discovery of certain nematode species thriving in anoxic conditions thousands of meters below the Earth's surface, in fractures within ancient rock formations. In the depths of South African gold mines, some reaching nearly 4 kilometers below ground, scientists have identified nematode species, such as Halocephalus mephisto, colloquially dubbed the worm from hell, living in water-filled fissures where oxygen is virtually non-existent. These subterranean waters are often ancient, geothermally heated, and rich in dissolved minerals, but critically lacking in the oxygen that surface-dwelling life depends upon. The presence of multicellular animals in such an extreme, isolated biome was a startling revelation. These deep-earth nematodes have clearly evolved remarkable adaptations to survive in this challenging milieu. Their metabolism must be capable of functioning anaerobically, perhaps relying on fermentation pathways similar to those seen in yeast or other, more specialized anaerobic respiratory chains that utilize alternative electron acceptors instead of oxygen, such as sulfate or iron. The exact biochemical mechanisms are still a subject of intense research, but their mere existence proves that animal life can persist in the deep, dark, anoxic subsurface. The energy sources for these subterranean ecosystems are also a point of interest. Without sunlight for photosynthesis, life in these deep biospheres often relies on chemosynthesis, where microbes derive energy from chemical reactions involving inorganic compounds, such as methane or sulfur compounds present in the rock and water. The nematodes, in turn, likely feed on these chemosynthetic bacteria, forming a unique food web entirely independent of solar energy and atmospheric oxygen, a truly alien ecosystem hidden beneath our feet. The discovery of these deep-dwelling nematodes has significant implications, not only for understanding the limits of life on Earth, but also for the search for extraterrestrial life. If multicellular organisms can survive in such extreme, oxygen-deprived conditions deep within our own planet, it raises the possibility that similar life forms could exist in subsurface environments on other planets or moons, where surface conditions might be too harsh. These tiny roundworms, masters of the anoxic underworld, expand our conception of the habitable zones within our planet and beyond. Returning to the more familiar realm of freshwater ponds and streams, we encounter the hydra, a small, simple invertebrate predator belonging to the same phylum as jellyfish and corals, the Cnidaria. While not strictly anaerobic in the sense of living entirely without oxygen for extended periods, hydra possess a remarkable ability to tolerate and survive in environments with significantly reduced oxygen levels, a condition known as hypoxia. This resilience is achieved through a fascinating capacity to dramatically reduce its metabolic rate, essentially entering a state of suspended animation. Hydra have a very basic body plan, consisting of a tubular body with a foot at one end for attachment and a mouth surrounded by tentacles at the other, used for capturing small prey like Daphnia. They lack complex respiratory organs like gills or lungs. Gas exchange occurs directly across their thin body wall, which is only 2 cell layers thick. This simple structure, while efficient in well-oxygenated water, can become a liability when oxygen levels drop, yet hydra have evolved to cope. When faced with hypoxic conditions, hydra can slow down their physiological processes to a crawl. Their movement becomes sluggish, their feeding rate plummets, and their overall energy expenditure is drastically curtailed. This metabolic depression allows them to conserve their internal energy reserves and minimize their oxygen demand, enabling them to survive for considerable periods until oxygen levels in their environment improve. It's a strategy of patience and endurance, waiting out the unfavorable conditions rather than attempting to maintain full activity. This ability is particularly important in their natural habitats, such as stagnant ponds or densely vegetated areas, where oxygen concentrations can fluctuate dramatically due to factors like temperature changes, organic decomposition, or algal blooms. By being able to downregulate their metabolism, hydra can persist in these variable environments where other, more oxygen-dependent organisms might struggle or perish. Their regenerative capabilities, for which they are famous, also play a role, allowing them to repair any potential cellular damage incurred during periods of stress. The study of hydra's response to hypoxia provides valuable insights into cellular survival mechanisms under low oxygen stress, which has relevance for understanding conditions like ischemia, restricted blood flow, in more complex animals, including humans. These seemingly simple creatures, with their minimalist approach to life, demonstrate a sophisticated adaptation to one of the most fundamental environmental challenges, showcasing how even basic organisms can evolve elegant solutions to survive in a world of fluctuating resources. Their quiet resilience is a testament to the efficiency of simplicity. We now delve into one of the most ancient lineages of multicellular animals on Earth, the sponges. These seemingly simple filter feeders belonging to the phylum Porifera have graced our planet's oceans for hundreds of millions of years, predating most other animal groups. Their evolutionary success is partly due to their remarkable adaptability, which includes the ability to survive in environments with extremely low or even temporarily absent oxygen levels. This is achieved primarily by drastically slowing down their metabolic processes. Sponges are sessile organisms, meaning they live attached to a substrate, and they feed by drawing water through their porous bodies and filtering out microscopic food particles. However, some sponge species, particularly those found in deeper waters, in enclosed marine caves, or in areas prone to seasonal oxygen depletion, have demonstrated an extraordinary capacity to endure anoxia for extended periods, sometimes for days or even weeks. When confronted with a lack of oxygen, these resilient sponges enter a state of metabolic arrest. Their ciliated cells, which normally beat to create water currents, may cease or significantly reduce their activity. Cellular respiration slows to a near standstill, and overall energy consumption plummets. It's a profound physiological shutdown, allowing the sponge to conserve its resources until oxygenated water returns. This is not a mere tolerance but a deep, evolved adaptation to survive the unpredictable nature of their environment. The cellular mechanisms underlying this anoxic survival are complex and involve coordinated downregulation of non-essential metabolic pathways while protecting vital cellular functions. They may rely on stored energy reserves, such as glycogen, and potentially switch to limited anaerobic pathways to generate ATP. The ability to recover fully once oxygen is restored is also crucial, indicating robust mechanisms for cellular repair and reactivation. The ancient heritage of sponges may offer a clue to their anaerobic capabilities. They evolved when Earth's oceans contained significantly less oxygen than today. It's possible that these early sponges evolved metabolic flexibility that allowed them to cope with fluctuating oxygen levels, an ancestral trait retained in certain modern lineages. These living fossils continue to surprise us with their resilience, demonstrating that even the simplest-looking animals can harbor sophisticated survival strategies honed over geological time. Our journey now takes us to some of the planet's most saline environments, salt lakes and evaporation ponds, home to the remarkable brine shrimp, scientifically known as. These small crustaceans are famous for their ability to thrive in extremely salty water, where few other creatures can survive, but also for their extraordinary reproductive strategy involving dormant cysts that can endure incredibly harsh conditions, including prolonged periods without oxygen. Often inhabit ephemeral waters that can dry up completely. To cope with this, female brine shrimp can produce tiny, hardy cysts instead of live young. These cysts are metabolically inactive embryos encased in a protective shell. In this state of cryptobiosis, similar to that seen in tardigrades, their life processes are almost entirely suspended, allowing them to withstand desiccation, extreme temperatures, and anoxia. These cysts can remain viable in anoxic sediments or dry salt flats for many years, sometimes even decades. Deprived of oxygen and water, they are in a profound state of dormancy, waiting patiently for conditions to become favorable again. When rainwater eventually floods their habitat, rehydrating the cysts, they can hatch within a few hours, releasing Nauplius larvae that rapidly develop into adult brine shrimp, restarting the life cycle. This ability to time travel through periods of adversity is a key to their survival. The anoxic survival of cysts is a marvel of biochemical engineering. The cyst shell itself provides a barrier, but the embryo within has its own protective mechanisms. They accumulate protective molecules like trehalose sugar and heat shock proteins, which help stabilize cellular structures during the dormant phase. Their metabolism is so profoundly suppressed that oxygen demand is virtually nil, allowing them to persist in buried, oxygen-free layers of sediment for astonishingly long durations. The resilience of brine shrimp cysts has made them valuable in aquaculture, where they are sold as sea monkeys or fish food, easily hatched on demand. Beyond their practical uses, provide a fascinating model for studying cryptobiosis. Their ability to package life into such a resilient, oxygen-independent form showcases yet another ingenious strategy evolved by nature to ensure continuity, even when the environment becomes utterly inhospitable, a testament to life's enduring power to wait for its moment. We return to the vast expanse of the ocean, but this time we explore environments known as oxygen minimum zones, or OMZs. These are regions in the water column, typically found at mid-depths, where oxygen concentrations are naturally very low, often too low to support most fish and other actively respiring marine animals. Yet within these challenging hypoxic waters, certain species of jellyfish not only survive, but can sometimes thrive, forming dense aggregations. Their ability to tolerate these conditions gives them a distinct advantage. Jellyfish, with their gelatinous bodies composed mostly of water and relatively low metabolic rates compared to more active swimmers like fish, are inherently better suited to low oxygen environments. Their simple body plan, lacking complex respiratory organs, means they rely on direct diffusion of oxygen across their body surfaces. While this might seem limiting, their large surface area to volume ratio, especially in larger individuals, can be surprisingly efficient for gas exchange, even when ambient oxygen levels are poor. Furthermore, some jellyfish species have evolved specific physiological and behavioral adaptations to cope with hypoxia. They may exhibit reduced activity levels, slowing their bell pulsations to conserve energy when oxygen is scarce. Some can also tolerate a buildup of anaerobic metabolic byproducts in their tissues for short periods. There is evidence that certain species can even engage in limited anaerobic metabolism to supplement their energy needs when oxygen is insufficient for fully aerobic respiration, allowing them to persist where their predators or competitors cannot. The existence of OMZs is a natural phenomenon, but these zones are expanding in many parts of the world's oceans due to climate change and nutrient pollution, leading to concerns about their impact on marine ecosystems. The ability of some jellyfish to tolerate and even flourish in these expanding low-oxygen areas means they may become increasingly dominant in certain regions. This can have cascading effects on food webs, as jellyfish are voracious predators of small fish larvae and zooplankton, and are themselves prey for only a few specialized animals. The resilience of these jellyfish in hypoxic waters is a stark reminder of how life adapts to fill every available niche, even those that seem marginal or hostile. Their ethereal, drifting forms, pulsing silently through the oxygen-poor twilight zones of the ocean, represent another facet of nature's ingenuity. Studying their adaptations can help us understand how marine ecosystems might shift in response to ongoing environmental changes, and how some life forms are pre-adapted to thrive where others falter, painting a complex picture of survival in the ocean's hidden depths. Our final exploration into the world of oxygen-independent survival takes us to the often overlooked habitats of anoxic muds and sediments found at the bottom of ponds, lakes, estuaries, and even the deep sea. Here, amidst the decaying organic matter and devoid of light and oxygen, thrive certain species of flatworms belonging to the phylum Platyhelminthes. These seemingly delicate, soft-bodied creatures have mastered the art of living where oxygen is but a distant memory, employing sophisticated alternative metabolic pathways. Many free-living flatworms, such as some turbellarians, are well adapted to these oxygen-depleted environments. Decomposition of organic material by bacteria in these muddy substrates rapidly consumes any available oxygen, creating permanently anoxic conditions just a few millimeters below the sediment surface. Flatworms inhabiting these zones cannot rely on aerobic respiration and have instead evolved the capacity for sustained anaerobic metabolism, primarily through various forms of fermentation. Their anaerobic pathways are often more complex and versatile than the simple lactate fermentation seen in vertebrate muscle during short bursts of activity. These flatworms can excrete a variety of end products from their anaerobic metabolism such as acetate, propionate, and succinate. This metabolic flexibility allows them to extract more energy from their food reserves under anoxic conditions than would be possible with simpler fermentation processes, enabling them to remain active, feed, and reproduce in the complete absence of oxygen. The ability to thrive in anoxic sediments offers flatworms a refuge from many predators and competitors that are dependent on oxygen. They glide through these dark, sulfurous muds, feeding on bacteria, detritus, or smaller invertebrates that are also adapted to this challenging environment. Their flattened bodies provide a large surface area for the absorption of dissolved organic molecules directly from their surroundings, supplementing their diet and further aiding their survival in these nutrient-rich yet oxygen-poor niches. The diverse metabolic strategies employed by these anoxic-dwelling flatworms highlight the incredible biochemical plasticity of life. They demonstrate that complex, multicellular organisms can evolve to uncouple themselves entirely from the need for atmospheric oxygen, forging a successful existence using ancient metabolic routes. These humble creatures, often hidden from our view, are yet another testament to the pervasiveness of life and its capacity to colonize even the most extreme corners of our planet, mastering the chemistry of survival in a breathless world. We have journeyed through realms unseen, from the tissues of a salmon to the abyssal depths of the ocean, and into the very fabric of the Earth's crust, encountering creatures that defy our most fundamental assumptions about life. The Henaguya, the Loricifera, the indomitable tardigrade, and their fellow masters of anoxia each tell a story of incredible evolutionary innovation. Their ability to thrive without oxygen, the element so central to our own existence, showcases the sheer breadth of biological possibility and the relentless drive of life to adapt and persist, no matter how challenging the environment. These organisms are not mere oddities, they are living proof of nature's profound creativity. From re-engineered cellular machinery to states of suspended animation and metabolic pathways that hark back to an earlier, oxygen-poor Earth, each adaptation is a masterpiece of survival. They remind us that our planet still holds countless secrets, and that the tapestry of life is far richer and more varied than we often appreciate. The capacity to live without air opens up entirely new perspectives on what it means to be an animal, and where life itself might be found. The stories of these 10 remarkable animals are but a glimpse into the extraordinary adaptability that life possesses. The natural world is a continuous source of wonder, a place of endless discovery. If you've been as captivated by these breathless wonders as I have, do consider subscribing for more explorations into the mind-blowing intricacies of the animal kingdom. Because as we have seen time and time again, nature always finds a way.